Okay. Okay. I will start now. The next we are going to talk about the cell signaling. Okay. Uh, all the cells receive and respond to the signal from their surrounding. For example, like a bacteria can sense and swim towards the high concentration of the nutrient. And even in the yeast cell, which is eukaryote, okay, during the condition or the situation like a mating, they have to communicate to another yeast cell that another yeast cell are ready for mating or not by secretion the protein or the signaling molecule to inform another yeast cell. That signaling molecule will bind to the receptor of another yeast cell. If they can bind and another cell recognize that another cell is the opposite sex, so that the mating will occur. I will show you the diagram or the picture, okay, showing the communication between yeast cell during the mating situation. For example, in this picture, there are two yeast cells. We call the A cell, which are the sex one, and another yeast cell we call alpha cell. Okay, it's supposed to have the opposite sex to the A cell. Okay, the A cell will release the A factor, which is like a signaling molecule. Okay, this signaling molecule will go to the B yeast cell, go to the alpha yeast cell, sorry. And if the alpha yeast cell have a receptor for the A factor, okay, the A factor can bind to the alpha yeast cell, and then the alpha yeast cell can recognize that another yeast cell is the opposite sex. Okay, at the same time, the alpha yeast cell will release the alpha factor, just in case to communicate another yeast cell, just to check that another yeast cell is the opposite sex of of itself or not. And after they found that they are the opposite sex, so the mating can occur, and then they will get the diploid cell, and then this is the seem like the cycle. For the multicellular organism, which is quite sophisticated and it's a complicated situation. So for the cell-cell communication is quite difficult when compared to the unicellular organism. So the communication usually sophisticated and has to carefully regulated to meet the need of the organism as a whole. For the communication between cells in the multicellular organism, they can use by many means or by many methods. They can use the direct contact, okay, to communicate the nearby or the neighboring cells. Or if the cell they want to communicate is far away, they can use the signaling molecule. This is the example of the communication of the cell that located nearby. Okay, we have the structure called gap junction in animal cell, as we learned it in the morning. Okay, and also the structure we call plasmodesmata in the plant cell. Okay, these two structures allow the communication between two cells, which located nearby. Okay, however, sometimes they use like a cell surface protein in order to communicate each other, like in this diagram. Okay, we call cell-cell recognition by using cell surface contact. The one used to the one cell has to have the like a specific protein, and another cell has to have the receptor. Okay, therefore, the cell recognition, they use like a receptor and protein, surface protein in order to communicate each other. But this process will be, efficient to communicate in the case that these two cells are located, they are very close. But in the case that the cell, they want to communicate, one cell want to communicate to another cell which is far away. So they have to use the signaling molecule. Okay, once the signaling molecule go to the target cell, okay, and alive reach at the target cell, that signaling molecule will bind to the receptor on the target cell. For example, like in this diagram, okay, this is a signaling molecule. And then when the signaling molecule bind to the receptor can be on the surface of the target cell or inside the target cell. After binding, okay, there is a complex we call receptor ligand binding. And after that, there is the there is a series of intracellular interaction, or we call signal transduction. This is the signal transduction pathway, okay, occur inside the target cell. And finally, the outcome is the response of the target cell, okay. The response of the target cell can be the expression of the specific protein or the activation of cellular response or activation of some specific enzyme inside the target cell. The cellular behavior that can be occurred after the signal transduction can be the change in the metabolism, or the change in the movement, or the change in the proliferation of the target cell. Today, my outline on the topic of cell signaling will be, we will learn about the signaling molecule and their receptor, what type of signaling molecule and what type of the receptors. The second one is we are going to learn the function of the receptor that located on the cell surface. The third topic will be the pathway of intracellular signal transduction. Okay, let's start the first topic, signaling molecule and their receptors. There are many types of signaling molecule. We can divide the signaling molecule into many types according to their structures. Okay, the signaling molecule can be as the gas or the molecule like protein. Okay, some of the signaling molecule can pass across the plasma membrane and act or bind to the receptor inside the target cell. However, some of the signaling molecule cannot pass across the plasma membrane. Therefore, they have to bind to the receptor located on the cell surface. And they have to have another additional pathway or mechanism to convey the information from the signaling molecule into the inside of the cell. This slide showing the mode of cell-cell signaling. Okay, it can be cell-cell interaction like this without any type of signaling molecule. And the communication between cells can be occurred by using the signaling molecule. Okay, we can divide the communication between cells using signaling molecule into three models. The first one we call endocrine signaling. The second one, paracrine signaling, and the third one, autocrine signaling. All of these communication pathway needs the signaling molecule. Okay, the differences is in detail of sending the signaling molecule. For endocrine signaling, like in this picture, okay, the important detail of endocrine signaling is that the signaling molecule has to be transported by the circulation. Okay, after releasing from the synthesized cell, the signaling molecule will go to the target cell by using the circulation system and arrive at the target cell. But this is endocrine signaling. But for paracrine signaling, the signaling molecule will go to the target cell without circulatory system involved. Okay, they can use the process called diffusion. So it means that the target cell is not far away from the synthesized cell. Okay, and autocline signaling means the communication, they communicate with itself. So the signaling molecule after produced and released from one cell will return back to bind to the receptor on the same cell. Okay, this is the detail about the communication between cells by using direct communication or direct interaction. In this picture, we give the example by using the cadherin. Okay, we learned in the morning that the cadherin is the protein, okay, transmembrane protein, and they need the divalent cation in the binding interaction. Okay, this cadherin molecule has it is the polypeptide, right? And this is the N-terminal of cadherin. The C-terminal is the part of the cadherin molecule that bind to cytoskeleton of the cell, okay, and also bind to actin filament, the red one, okay, the red one. Okay, and also cadherin has a transmembrane domain and also extracellular domain here. Okay, because this cadherin bind to the actin filament, therefore we call this cadherin, we call ethylene junction, right? Because it binds to the actin filament. And this cadherin, okay, they also serve to regulate the cell proliferation and survival. These are the details of communication by using the signaling molecule. The first one is endocrine signaling, or we call hormone. Okay. The specificity of endocrine signaling is that the signaling molecule has to be transported to the target cell by circulation. Okay, and nowadays we have already known the hormone, more than 50 different hormones that is the endocrine signaling. For paracrine signaling, okay, the transportation of signaling molecule to the target cell occurred by diffusion. The example of signaling molecule in this paracrine signaling is neurotransmitter. So it means that neurotransmitter can be released from one nerve cell, okay, and diffuse across the synapse to another nerve cell or to the muscle cell, okay, in the neuromuscular junction. For autocline signaling, okay, the example of autocline signaling is the response to the signaling molecule they produce themselves. For example, like a T lymphocyte synthesize growth factor, okay, and release and control the proliferation of the nearby cell together with their own proliferation. This is the picture showing endocrine signaling, okay? Signaling molecule produced, get into the circulation, transported in the circulatory system, and go to the target cell. Paracrine signaling from the synthesized cell, diffusion, and then bind to the receptor on the target cell. Autocrine signaling produced and return back to bind to the receptor on the same cell. Okay, this is the example of paracrine signaling. Okay, it can be from the synthesized cells, released secretary, released from a signaling molecule released from the secretory vesicle, and diffusion go to the target cell nearby, or in the nervous system. Okay, this is also called paracrine signaling. Okay, the signaling molecule released from the axon of the nerve cell and then go to another nerve cell, okay, across the synapse to bind to the receptor on another nerve cell. Autocrine signaling. Okay, so from now on, we are going to learn about the signaling molecule. Okay, there are many types of signaling molecule. We start from the signaling molecule that have the structure of steroid. Okay, so we call steroid hormone. Okay, the signaling molecule like a steroid hormone has a structure, I mean, non-polar molecule. Okay, this molecule which have hydrophobic properties can passively diffusing across the plasma membrane because they have no polar and also they have no charge. Therefore, they can go across the plasma membrane and bind to the intracellular receptor. Okay, the signaling molecule that have these the same properties like this is the steroid hormone, thyroid hormone, vitamin D, and also retinoic acid. Okay, this slide shows the structure of steroid hormone. This is the common structure of steroid hormone. This is thyroid hormone, and this is vitamin A. Okay. Okay, look at the thyroid hormone. Okay, even if the thyroid hormone is not steroid hormone, however, the thyroid hormone is quite small. Therefore, they can pass across the plasma membrane and go inside the cell and bind to the receptor that located in the nucleus of the target cell. Okay, the member of steroid hormones are testosterone, estrogen, progesterone, glucocorticoid, mineralocorticoid. All of the steroid hormones have the same precursor. Okay, they synthesize the starting molecule cholesterol. Okay, this is the structure of cholesterol. Cholesterol have a 4 ring. Okay, we call A, B, C, D. Okay, the differences between these steroid hormone, they share the common structure like a B, C, D ring. The differences between these steroid hormone are the side chain. Okay, you can look at the differences in the side chain. And also the steroid hormone receptor. Okay, steroid hormone receptor is the receptor that located inside the target cell. Okay, the receptor are protein. Okay, and the common structure of steroid hormone receptor, they have to have 3 domains. The first domain we call ligand binding domain of the receptor. Okay, the receptor use this domain to bind to the steroid hormone. The second domain is the DNA binding domain. The receptor use this part of the receptor, okay, this part of the structure to bind to the DNA. And the third domain is the transcriptional activation domain. Okay. So if we look at the structure of estrogen, progesterone, glucocorticoid receptor, okay, these three receptors are the receptor of steroid hormone. Okay, you can see that there are differences in the length, I mean the size of the receptor. However, they share the same structure, that they have the ligand binding domain in the blue color. Okay, and they also have the DNA binding domain in the pink color. Okay, together with thyroid hormone receptor, they also share the ligand binding domain and DNA binding domain. And also, this is also the last one is the receptor for the vitamin A. So when you see the structure of the receptor, you can guess that this receptor will definitely bind to the DNA and stimulate or regulate the DNA expression, because they have the domain that bind to the DNA. So it means that after this receptor binding to the ligand receptor complex will go to the DNA and bind to the DNA and stimulate the change in the DNA expression. Okay, there are two types of intracellular receptor. Okay, the first one is the receptor that normally not bind to the DNA. This is the receptor of estrogen and glucocorticoid. Okay, you can see from the diagram, from the picture here, this is the receptor. Okay, normally when there is no ligand, this receptor is free, okay, and located inside the nucleus. Okay, and when there is the steroid hormone get into the cell and pass through the nuclear membrane, okay, this steroid hormone will bind to the receptor, and after binding, there is the receptor ligand complex. Okay, this is they become the receptor by ligand complex, and after that, the conformation of the receptor change after binding to the ligand, and this conformation can fit to the DNA, and they can go to the DNA and bind to the DNA and stimulate the transcription and activate the transcription. Okay, another type of intracellular receptor is the receptor that already bound to the DNA, but they didn't do anything with the DNA, just only bind to the DNA. Okay, for example, like in this picture, in the absence of hormone, okay, the receptor just only bound to the DNA, but there is no transcription, no expression of the DNA. But when the hormone, okay, get into the cell and bind to the receptor, the binding cause the conformational change of the receptor, and this conformational change can stimulate or can activate the transcription. Okay. The example of this receptor is the receptor of thyroid hormone. The second group of signaling molecule is gas. Okay. The example is nitric oxide gas or carbon monoxide gas. Nitric oxide gas is the major paracrine signaling molecule in nervous, immune, and circulatory system. Of course, nitric oxide gas is very, very small, right? The size is very small, and also it is gas. Therefore, nitric oxide gas can diffuse freely directly across the plasma membrane. However, the action of nitric oxide is not bind to the receptor intracellularly, but nitric oxide have action on the enzyme. Nitric oxide will alter or change the activity of intracellular target enzyme, like the enzyme that change from GTP to cyclic GMP, guanylyl cyclase. Nitric oxide is the gas produced from arginine. Okay, the substrate of nitric oxide or the precursor of nitric oxide is arginine, amino acid arginine. By using the enzyme called nitric oxide synthase, arginine change to nitric oxide. And after that, nitric oxide will diffuse out of the synthesized cells and act locally. Because nitric oxide have a short half-life, just only 2 or 3 seconds. Therefore, the nitric oxide just act locally and regulate the nearby cell. I mean, the target cell of nitric oxide is just close to the synthesized cell of nitric oxide because half-life is quite short. The action of nitric oxide is to dilate the blood vessel. Okay, we can see the mechanism of synthesizing and also the mechanism of nitric oxide on regulating the smooth muscle. Okay, to synthesize nitric oxide is begin with the releasing of neurotransmitter acetylcholine at the end of the nerve cell in the blood vessel wall. Okay, acetylcholine, after release from the nerve ending of the blood vessel wall, acetylcholine will stimulate the synthesis of nitric oxide from arginine in the endothelial cell. And then, okay, I will let you see here. Okay, acetylcholine, okay, will stimulate the endothelial cell, okay, and activate the synthesis of nitric oxide from L-arginine by nitric oxide synthase enzyme. Okay, this is the calcium ion is necessary, okay, as a cofactor for nitric oxide synthase. And after that, the product is nitric oxide. The nitric oxide will diffuse across the plasma membrane and get out from the synthesized cell, endothelial cell, and go to the smooth muscle cell. And in the smooth muscle cell, nitric oxide will stimulate the guanylyl cyclase enzyme. The guanylyl cyclase enzyme is the enzyme that helps to change from GTP to cyclic GMP. And the cyclic GMP is the second messenger and can go to and can stimulate the relaxation of the smooth muscle. And finally, the result is the dilation of the blood vessel. Okay, this this is how the nitric oxide can be synthesized from L-arginine. This is the structure of L-arginine. Okay, and by the help of nitric oxide synthase and also the reducing equivalent NADPH, the product is nitric oxide together with another type of amino acid we call L-citrulline. Okay. The second group of signaling molecule is neurotransmitter. Okay. The first is steroid hormone. The second is the gas, nitric oxide. And the third group of signaling molecule is neurotransmitters. Okay, the neurotransmitter carry the signal between neuron or between neuron and another type of target cell like a muscle. Neurotransmitter released from the terminus of the neuron. Okay, upon the arrival of action potential along the nerve fiber. Okay, and then the neurotransmitter is released. Neurotransmitter molecule diffuse across the synapse and bind to the receptor on another neuron. Okay, like this one. Okay, because of the action potential, they stimulate the release of neurotransmitter. And once the neurotransmitter release and go across the synapse to the another neuron target cell and bind to the receptor on the target neuron cell. The neurotransmitter is the small hydrophilic molecule. The example of neurotransmitters are acetylcholine, dopamine, epinephrine, serotonin, histamine, glutamate, glycine, and gamma aminobutyric acid or GABA. Okay, all of these neurotransmitters has either polar or charge. Therefore, they cannot cross the plasma membrane freely. That means they have to bind to the receptor located on the surface of the target cell. So they have to bind to the cell surface receptor. Okay, the neurotransmitter can bind to the receptor on the surface of the target cell. Some receptor is the ion channel itself. So it means that besides performing as a receptor, they also has a properties of ion channel, or we call ligand gated channel. For example, like in this picture. Okay, this is the receptor of neurotransmitter. Neurotransmitter is the green ball. After binding to the receptor itself is the ion channel. Therefore, after binding, they change the conformation, the binding between signaling molecule neurotransmitter and the receptor create the conformational change of the receptor, which is the ion channel 2. So then the ion channel open because of conformational change, and then the ion can get into the target cell. Some receptor, like this one, is not the ion channel itself, but they can create, okay, they can send the information to the ion channel nearby, okay, and regulate the opening of the ion channel nearby, and then the ion can influx into the target cell. Okay, in this process, the receptor of the neurotransmitter stimulate the conformational change of the nearby protein, for example, like a G protein. And after G protein is stimulated, is activated by the ligand receptor complex, the activated G protein can regulate the opening of the ion channel nearby. Some neurotransmitter can act as the hormone, for example, like epinephrine. Okay, as you know that epinephrine has two functions. Okay, the first one is a function as neurotransmitter in the sympathetic nervous system. Okay, and the second function of epinephrine is the hormone. Okay, synthesized from the medulla of adrenal gland, or we call it, and also epinephrine is the member of the group of the hormone called catecholamine. The epinephrine act as the hormone, okay, can signal the glycogen breakdown in the liver cell or in the hepatocyte. Okay, this is the action of epinephrine as the hormone. Okay, this is the picture showing the action of epinephrine and glucagon on the hepatocyte or on the liver cell. Okay, after binding to the receptor, there is the change in the hepatocyte. Okay, that is the process called gluconeogenesis and also glycogenolysis. And finally, there is the release of free glucose from the liver cell into the circulation. And we get the increase in the glucose concentration in the circulation after the action of epinephrine and glucagon. The fourth group of the signaling molecule is the peptide hormone and growth factor. Okay, growth factor is also protein. This group of signaling molecule is the biggest group of the signaling molecule in the animal. Okay, they are composed of peptide hormone, neuropeptide, okay, polypeptide, growth factor. This type of signaling molecule are quite big in size, and they cannot cross the plasma membrane. Therefore, they must bind to the receptor located on the cell surface of the target cell. The example of peptide hormones are insulin, glucagon, growth hormone, follicular stimulating hormone or FSH, and also prolactin. All of them are peptide hormone. Okay, neuropeptides are enkephalin or endorphin. Okay, this is the product of the the neural cell or neuron. Instead of produce just only neurotransmitter, the neuron also can produce like a peptide. For example, enkephalin or endorphin. Enkephalin and endorphin or neuropeptides can have a function as neurotransmitter and act at the neuron nearby. For example, like across the synapse and bind to the receptor on the neighboring neuron. Okay, or can get into the circulation and act as a neurohormone. There are many There are many types of polypeptide growth factors. The first one is a nerve growth factor. This polypeptide growth factor has a function as controlling animal cell growth and differentiation. Like in nerve growth factor or NGF, the nerve growth factor is a polypeptide or protein. And this protein regulates the development and survival of the neuron. Epidermal growth factor. These growth factors stimulate the cell proliferation. And it is composed of 53 amino acids. Okay. This diagram this picture show the epidermal growth factor and also their receptor. Okay, this is a receptor of epidermal growth factor on the cell surface of the target cell. The third type of growth factor is pellet-derived growth factor. This growth factor is stored in the blood pellet and released during the period of blood clotting at the site of the damaged tissue or wound. And also this growth factor will stimulate the proliferation of fibroblasts in order to stimulate the regrowth of the damaged tissue. Cytokine. Cytokine is the product from the from many cells, including the immune cell. Cytokine can regulate the development and differentiation of blood cell and also control the activity of the immune cell like a lymphocyte during the immune response. Okay, in this picture, you can see many types of cytokine like a interleukin 12, okay, interleukin 4, and also interleukin 4, 5, 6, tumor necrosis factor, and also gamma interferon. They are all cytokines. And this is also the quite complicated diagram, okay, and show the releasing of cytokine, releasing of interleukin, many types of interleukin to control all the system of the immune cell. Another type of growth factor we call membrane ensure growth factor. This growth factor is quite different from the growth factor that I mentioned before, because this growth factor is not the soluble one. Okay, this growth factor is located on the cell membrane. That's why we call membrane ensure growth factor. They associated with the plasma membrane and function as the signaling molecule during the direct cell-cell interaction. So they communicated with the nearby target cell. Another type of signaling molecule is the lipid. From the lipid structure of the signaling molecule. Okay, we call eicosanoid. Okay, this lipid, this signaling molecule comes from lipid, but different from steroid hormone. Different from steroid hormone because this signaling molecule doesn't have the precursor as cholesterol. That's why they different from the steroid hormone. Okay, the precursor of this signaling molecule is the fatty acid. Okay, this is the fatty acid, like a arachidonic acid. Okay, we call this signaling molecule eicosanoid. Okay, the example or the member of eicosanoids are prostaglandin, prostacyclin, thromboxane, and leukotriene. Okay. Most of them has the same precursor, arachidonic acid, okay, which is the phospholipid and found as the composition of plasma membrane. During the specific situation, for example, like a inflammation, infection, injury, the phospholipid can release from the cell membrane and become free phospholipid and act as the precursor for the synthesizing eicosanoid. Okay. In this diagram, the arachidonic acid is released from phospholipid to become free arachidonic acid. And this arachidonic acid acts as the substrate for the enzyme cyclooxygenase. Okay, cyclooxygenase is the enzyme that produces the product from arachidonic acid. For example, like a prostaglandin, thromboxane, okay, and the drug like a NSAID can inhibit the action of cyclooxygenase, so inhibit the synthesis of eicosanoid from arachidonic acid. Together with some steroid drug like dexamethasone also can inhibit the synthesizing eicosanoid by inhibiting the action of cyclooxygenase enzyme. There are two types of eicosanoid. We make it easy to call it like a good eicosanoid and bad eicosanoid. Good eicosanoid means the eicosanoid that prevent the damage of the cell, and also the eicosanoid that inhibit the inflammation. However, the bad eicosanoid means the eicosanoid that stimulate the damage of the cell and also activate the inflammation in the tissue. Aspirin is the drug that already we know it for many years. Aspirin is one of the NSAID group. Okay, aspirin can inhibit the enzyme cyclooxygenase, meaning that aspirin can reduce the inflammatory reaction and also reduce pain. And also aspirin can reduce platelet aggregation and blood clotting. Besides, aspirin can prevent stroke, can reduce frequency of colon cancer. Why? This is because aspirin can inhibit the production of eicosanoid, right? As we know from the last slide. And the product of one of the eicosanoid product is prostaglandin from arachidonic acid. We know that prostaglandin can stimulate the cell proliferation and promote the cancer development. Therefore, if we can inhibit the synthesis of prostaglandin, therefore we can slow down, or we can prevent, or we can inhibit the development of cancer. This is why nowadays in the medicine we use NSAID in the cancer condition, just to slow down or to prevent or partially inhibit the production of prostaglandin, which is the key molecule that stimulate the cell proliferation in the cancer. The next one is the function of cell surface receptor. Most of the ligand that I mentioned before, or ligand or is the same as signaling molecule. Okay, most of the signaling molecule, for example, like neurotransmitter, peptide hormone, and also growth factor, is a large group of the signaling molecule. All of these molecules cannot pass through the plasma membrane and get into the target cell. Therefore, they have to bind to the surface receptor on the target cell. For the neurotransmitter receptor, okay, neurotransmitter receptor, mostly this type of receptor is the ion channel itself. Therefore, after binding, that is the control of ion influx across the membrane. But for the peptide, hormone, and also growth factor, the receptor of the peptide, hormone, and growth factor usually will regulate the activity of intracellular proteins, for example, like intracellular enzyme. Okay, when the ligand bind to the cell surface receptor, okay, that is the mechanism or process occurred inside the cell. Okay, this is called intracellular reaction. This reaction is the chain reaction. So it means that there is a cascade reaction occurred after the ligand or signaling hormone or signaling molecule bind to the receptor on the surface of the cell. Okay. And the reaction occurred in the cytoplasm and sometimes it occurred in the nucleus. And this complex, I mean, like a ligand and receptor complex or the signaling intracellular signal can change the gene expression program in the target cell. There are 4 types of cell surface receptor. Okay, the first one we call G protein coupled receptor. The second one is the receptor protein tyrosine kinase. The third type of cell surface receptor is cytokine receptor and non-receptor protein tyrosine kinase. And the fourth type of cell surface receptor is receptor linked to other enzymatic activities. Okay, we start from G protein coupled receptor. This is the largest family of cell surface receptor. Okay. G protein coupled receptor is the guanine nucleotide binding protein. Okay. G protein coupled receptor is the receptor of neurotransmitters, neurotransmitters, and also the receptor of neuropeptides, and also the receptor of peptide hormones. Therefore, that's why the G protein coupled receptor is the biggest cell surface receptor, because it is the receptor of almost the signaling molecule. Okay, this is the structure of G protein coupled receptor. Okay, this receptor is bind to the G protein. Okay, the blue one is the G protein, and G protein located intracellular side of the cell membrane. Okay, this is the intracellular side, and this is the extracellular side of the plasma membrane. This is G protein. Okay. Okay. So when we look at the G protein coupled receptor, we can see that this receptor span, okay, span the plasma membrane 7 times. Okay, or it can, this receptor passed, the structure of receptor passed through the plasma membrane 7 times. Sometimes we call this type of receptor 7-pass transmembrane receptor. Okay, and also G protein coupled receptor is also the receptors of smell, sight, and taste. This is G protein. Okay, G protein is composed of 3 subunits: alpha subunit, beta subunit, and gamma subunit. Okay, this G protein linked to the 7-pass receptor. Okay, how the G protein coupled receptor work? Okay, we can see the diagram. We can learn from the diagram. Okay, this is the G protein couple receptor or 7 parts. Okay, and this is the G protein. Okay, G protein has alpha subunit, beta subunit, and gamma subunit. At the resting state, no ligand bind. The G protein bind to the nucleotide GDP. Okay, and once there is a ligand bind to the receptor here, okay, there is a after binding, there is a conformational change of the receptor. And because of conformational change, this change induce or stimulate the G protein nearby. When the G protein is activated, they cannot no longer bind to the nucleotide GDP. They can bind to another type of nucleotide, T tower P. And once the alpha subunit of G protein bind to GTP, this alpha subunit become activated, and it can stimulate or induce the activation of other type of protein inside the target cell. After that, the G protein that bind to the activated alpha subunit will be hydrolyzed. And when GTP hydrolyzed, there is a GDP. So this GDP bind to alpha subunit, leading to the inactivation of the alpha subunit, and they go back to the resting state. There are many types of G protein couple receptor, okay, especially the type of G protein. If this G protein, after being activated by the binding between ligand and receptor, if this G protein stimulate the enzyme, the specific enzyme like adenylyl cyclase, we call this G protein GAs. But some G protein, after being activated, inhibit the guanylyl cyclase. This G protein we call GI. And some of G protein, after being activated, will regulate the ion channel or regulate the target enzyme. Okay. For example, in this picture, the G protein can be G inhibit GI, G stimulate like a GS, okay, and also G protein can involve in the production of second messenger like a DAG and IP3. That is we call GQ. This is the example of how G protein couple receptor work. Okay, we give the example of the action of acetylcholine as a neurotransmitter. Okay, this is the cell. Okay, on this cell they have two types of cell surface receptor. The first one is the receptor for epinephrine, and the second one on the right hand side is the receptor of acetylcholine. Okay, epinephrine will bind to the receptor. The receptor of epinephrine is G protein couple receptor, meaning that after binding between epinephrine and receptor, this ligand or hormone receptor complex will stimulate the change of G protein. And the G protein of epinephrine hormone is the G stimulate, GAs. So meaning that after binding of epinephrine to the receptor, there is the activation of GS alpha subunit, and they will stimulate the enzyme nearby, adenyl cyclase. And this enzyme, after activation, will involve in the change from ATP to cyclic AMP, meaning that epinephrine will create the increased concentration of cyclic AMP in the target cell. However, the acetylcholine, which is a neurotransmitter, okay, will bind to the receptor. Okay, the binding between acetylcholine and receptor will stimulate the G protein, but G protein in this condition, it is a GI. So it means that this G protein alpha subunit will inhibit the action of adenylate cyclase enzyme. And the inhibition of this enzyme result in the decrease of cyclic AMP. Or in other words, we can say that acetylcholine can have the final response of the target cell as the low concentration of cyclic AMP inside the cell. The second type of cell surface receptor is the receptor protein tyrosine kinase. Okay. This is the largest family of the cell surface receptor which have the intracellular enzyme as one of the receptor domain. This type of receptor is the receptor of almost polypeptide growth factor. Okay, these receptors share a common structural organization as N-terminal of these receptor is located extracellularly and bind to the ligand. And that is the single transmembrane alpha helix instead of 7. Okay, transmembrane helix is only one pass. And also another domain of this receptor is the cytosolic C-terminal domain. Okay, and at this part of the molecule, it is tyrosine kinase. They have tyrosine kinase activity. Okay. This is the structure of one pass, or we call receptor tyrosine kinase, receptor protein tyrosine kinase, which is this is the extracellular domain, right? And this is intracellular domain, and this is transmembrane domain. Okay, and the intracellular domain or cytoplasmic domain, this part of the receptor is tyrosine kinase. They have tyrosine kinase activity. This one. Okay, the one part of the receptor perform as a tyrosine kinase. This is the receptor of epidermal growth factor. This is a receptor of insulin, and this is the receptor of platelet-derived growth factor. Because you can see this single pass transmembrane protein. How this receptor function? Okay, the binding of the ligand. Okay, this is growth factor, right? The binding of the ligand to the receptor activate. Okay, the cytosolic domain, which is the enzyme. Because of the binding, we activate the enzyme from inactivate form to activate form. And because this is the protein tyrosine kinase enzyme, so it means that this enzyme can phosphorylate or add the as the phosphate group to another type of protein. Okay, in this picture, they add the phosphate, okay, to the another type of enzyme. Okay, we call PLC, gamma enzyme, phospholipase C gamma enzyme. Okay, and once this enzyme is phosphorylated, this enzyme become active and then can hydrolyze phospholipid PIP2, okay, to get two type of product. The first one is diacylglycerol, DAG, and the second product is inositol triphosphate, IP3. Okay. The phosphorylation, the phosphate comes from ATP, okay, and once the phospholipase C enzyme is phosphorylated, it becomes active. Okay, the third group of the cell surface receptor is the receptor of cytokine. And also this type of receptor has the protein kinase activity, not as the domain of the receptor, but the separate protein, but linked to the receptor. Okay. For example, this is the picture of type 3 cell surface receptor. It is also one-pass transmembrane receptor. However, the cytosolic domain of the receptor is not the enzyme itself, but the type of this type of enzyme is the separate protein that located link or join to the cytosolic receptor, cytosolic domain of the receptor. The example of this receptor is a receptor of almost cytokine. The fourth group of the cell surface receptor is the receptor that link to other type of enzymatic activity, not tyrosine kinase. For example, link to the another type of enzyme. For example, like a tyrosine phosphatase, okay, protein serine threonine kinase, or guanylyl cyclase. Okay. The third part of this lecture is the pathway of intracellular signal transduction. Okay. Most of the cell surface receptor stimulate intracellular target enzyme, either by directly link, okay, or indirectly coupled to the receptor by G protein. Okay. Intracellular enzyme, after being activated, okay, they can control the downstream pathway of the reaction in the target cell. This is the definition of intracellular signal transduction. It means the process of chain of reaction, okay, transmitting signal from cell surface to variety of intracellular target. For example, like a transcription factor. Therefore, there is a change finally in gene expression in order to respond to the extracellular stimuli. Okay, this is the example of intracellular signal transduction. The response is transcription. Okay. This is the example of ligand or signaling molecule, for example, like steroid hormone. In this example, they use testosterone as the signaling molecule. Testosterone is a steroid hormone that can pass through the plasma membrane. Therefore, they get into the target cell. And the receptor of testosterone is intracellular receptor. It is located in the cytoplasm. Okay. After testosterone bind to the receptor, we call the hormone receptor complex. Okay, this structure can get into the nucleus by passing through the nuclear membrane. And because of the binding between testosterone and receptor, create the conformational change of the receptor. This receptor hormone complex can then bind to the DNA and stimulate the DNA expression, stimulate the transcription. Then they get the messenger RNA here, and the messenger RNA can go out from the nucleus to the cytoplasm, and that is the protein synthesis pathway occurred in the target cell. Therefore, intracellular signal transduction of testosterone, that is the change in the DNA by stimulate or activate the DNA expression as transcription, and then followed by translation and the new protein synthesis. The intracellular signal transduction can be occurred by cyclic AMP pathway. Okay, for example, like the hormone, okay, and kind of ligand bind to the receptor, which is G protein couple receptor. After the binding, there is the activation of alpha subunit of G protein. The activated alpha subunit can stimulate the nearby enzyme called adenylyl cyclase. Once the adenylyl cyclase is activated, okay, there is a change from ATP to cyclic AMP. Finally, after the binding of the ligand to the receptor, there is the increase of cyclic AMP intracellularly. This is cyclic AMP. Okay, sometimes we call cyclic AMP as the messenger, because from now on, the pathway or the chemical reaction that occur in the target cell can be stimulated by the cyclic AMP. Therefore, we can call cyclic AMP as the second messenger. Cyclic AMP itself can be inactivated by the enzyme cyclic AMP phosphodiesterase. And then cyclic AMP become AMP, which no longer have the action. Okay. In the cyclic AMP signal transduction pathway in the target cell, okay, we give the example of epinephrine action. Okay, epinephrine will bind to the cell surface receptor, and because receptor of epinephrine is G-coupled receptor, G protein couple receptor, this binding will stimulate the G protein. Okay, and G protein of epinephrine is the GS stimulation. Therefore, the activated G protein will stimulate the action of adenylyl cyclase. Adenylyl cyclase will change from ATP to cyclic AMP. That is the increase of cyclic AMP in the target cell. The cyclic AMP can stimulate the protein kinase enzyme inside the target cell. This enzyme we call protein kinase A. And the protein kinase A will stimulate the phosphorylase kinase enzyme later. The phosphorylase kinase enzyme will phosphorylase and activate the glycogen phosphorylase, and finally that is the breakdown of glycogen. This is the action of cyclic AMP. And then there is the release of glucose from glycogen and release of the glucose from the cell into the blood circulation. Cyclic AMP can also induce gene expression too via the activated protein kinase A. Okay, I will show you the picture. This is protein kinase A, which is activated by cyclic AMP. The protein kinase A can go either this way or this pathway. The protein kinase A can phosphorylate many type of intracellular enzyme. For example, glycogen synthase enzyme. Without phosphate, this enzyme is active form. But when they bind to the phosphate, this enzyme is inactivated in the active form. Okay. However, if the protein kinase A acts as the enzyme to give the phosphorylation to the phosphorylase kinase enzyme, this enzyme becomes active with phosphorylation. And this active enzyme will phosphorylate another type of enzyme called glycogen phosphorylase. This is the enzyme. Normally it is inactive, but after being phosphorylated like this enzyme become active and can hydrolyze glycogen to become the glucose 1-phosphate and then free glucose in the liver, and then increase the glucose level in the blood circulation. This is the action of cyclic AMP after hormone binding to the receptor. When the cyclic AMP involved in the gene expression, okay, this mechanism occurred by cyclic AMP here, and then cyclic AMP has to stimulate, okay, the protein kinase A. Okay, this is protein kinase A. Normally, it is in an inactive form because the protein kinase A, okay, protein kinase A is bind to the blocker, bind to the R molecule, which is the blocker, and cause the protein kinase A inactive. Cyclic AMP will remove the blocker, will remove the R molecule. Without the R molecule, protein kinase A become active and then get into the nucleus. The protein kinase A, active protein kinase A, will phosphorylate the DNA binding protein. The DNA binding protein after phosphorylation is become active and stimulate the transcription. This is why cyclic AMP can also induce gene expression too, besides the activation of cytoplasmic enzyme. Cyclic AMP also the second messenger of ion channel open. Sometimes cyclic AMP functions alone, okay, directly regulate ion channel, independent of protein phosphorylation. For example, like in the sensing smell, okay, like in this diagram, okay, this is odorant, like a perfume, molecule from perfume will bind to the receptor, okay. This is the G protein couple receptor, and this G protein will activate the adenylate cyclase and get the high level of cyclic AMP, and this cyclic AMP will affect on the ion channel, okay, and that is the influx of sodium and also calcium ion in the target cell. About cyclic GMP pathway, in the cyclic GMP pathway, the second messenger is cyclic GMP. Okay, this is the cyclic GMP is important second messenger as cyclic AMP. However, the mechanism of second messenger cyclic GMP is not quite understood compared to the second messenger cyclic AMP. Cyclic GMP is synthesized from GTP by the enzyme guanylyl cyclase. And also, this is cyclic GMP. And also, cyclic GMP can be destroyed, okay, by the one specific enzyme called phosphodiesterase. And from cyclic GMP becomes 5' GMP with no more function. Cyclic GMP acts as the second messenger in the mechanism of nitric oxide. Okay, this is nitric oxide can pass through the plasma membrane, and nitric oxide has no receptor inside the target cell. Okay, nitric oxide can act on guanylyl cyclase, activate guanylyl cyclase, and then guanylyl cyclase activated guanylyl cyclase will change GTP to cyclic AMP, okay, to cyclic GMP, from GTP to cyclic GMP. And also cyclic GMP, similar to cyclic AMP, will stimulate the action of protein kinase G here. Okay, the action of protein kinase G after being activated. This activated protein kinase G will act on the smooth muscle relaxation, will act on the myosin phosphatase enzyme, and also the result of protein kinase G is pellet inhibition, and also change in gene expression. Cyclic GMP is the second messenger in vertebrate eye converting, okay, visual or light to the nerve impulse. Okay, this is the mechanism of cyclic GMP, act as the second messenger in the visual signal to create the nerve impulse and send to the brain that you can see the object. Okay, this is the effect and the role of cyclic GMP in how you can see the object. Another type of second messenger is the calcium ion. Okay. And also That is the two types of second messenger that synthesize from the plasma membrane phospholipid. The precursor of second messenger like a diacylglycerol and inositol triphosphate. The precursor is phosphatidyl inositol 4,5 biphosphate or PIP2. Okay, this phospholipid is located at the inner layer of the plasma membrane. That is the enzyme called phospholipase C that can hydrolyze PIP2. After being hydrolyzed, the product is diacylglycerol or DAG and inositol triphosphate or IPT. DAG and IP3 are second messenger. They can stimulate downstream signaling pathway, okay, via the protein kinase C calcium mobilization. This is the example of hydrolyzing phospholipid PIP2 in order to get second messenger like a IP3 and DAG. Okay. This mechanism, the ligand is growth factor, and of course the receptor is one part protein tyrosine kinase, meaning that the cytosolic domain of the receptor perform as the enzyme tyrosine kinase. That is the phosphorylation of the enzyme phospholipase C gamma here. And after being phosphorylated, this phospholipase C gamma become active. And then this enzyme will hydrolyze PIP2 and get two of the product DAG and IP3. These two substances is the second messenger for the intracellular signal transduction. There are two types of phospholipase C. Okay, they are phospholipase C beta and phospholipase C gamma. Phospholipase C beta is stimulated by G protein. However, phospholipase C gamma is stimulated by protein tyrosine kinase. Second messenger DAG from PIP2 substrate will associated with plasma membrane. Here, will associated with plasma membrane. Okay, and then activate another type of protein kinase which control the cell growth and differentiation. The substance called phorbol ester. This substance promotes the growth of tumor in animal by acting analog to DAG or DAG, second messenger, and stimulate the protein kinase C, okay, which then stimulate the expression of the gene. IP3 is another type of second messenger produced from PIP2 substrate. Okay, this IP3 molecule, this molecule is very small. We found this molecule in the cytosol. This molecule will stimulate the release of calcium ion from the storage inside the cell. Okay, in the diagram, this is PIP2 substrate, and because of phospholipase C hydrolyze PIP2, the product is, this is phospholipase C, okay? The product is IP3 inositol triphosphate, and DAG is here. IP3 small molecule will go to endoplasmic reticulum, which is the storage of intracellular calcium. IP3 will bind to the calcium ion channel on the cell surface on the plasma membrane of endoplasmic reticulum. This is endoplasmic reticulum, and this is plasma membrane of endoplasmic reticulum. This is the receptor, this is the calcium ion channel, and also the receptor of IP3. After IP3 bind to this receptor, there is a conformational change of this receptor, and also it is the ion channel itself. This ion channel is open, and that is the release of calcium ion from endoplasmic reticulum into the cytoplasm or into the cytosol. Therefore, after the binding of IP3 to the receptor, okay, to the receptor, there is the increase in the concentration of calcium ion in the cytoplasm of the target cell. This increased calcium ion will activate the protein kinase C together with DAG. Okay, after protein kinase C is activated, the active protein kinase C will phosphorylate another type of protein in the cytoplasm, and there is the cascade reaction, and finally the cell response of the target cell can be seen. Calcium ion is also the common second messenger, because intracellular calcium ion can affect the activity of many intracellular target protein. For example, like I mentioned before, that the calcium ion also together with DAG can activate the protein kinase C. And also another type of protein like calmodulin also needs calcium ion. The calcium concentration inside the cell can be increased by the extracellular calcium ion. Okay, extracellular calcium ion can enter the cell after the calcium ion channel open by the action potential or by membrane depolarization. The increase in the intracellular calcium ion can trigger the release of intracellular store. For example, in the lyanodine receptor, and then there is the release of neurotransmitter occur later. Second messenger from other phospholipid like DAG is also from phosphatidylcholine. Okay, we still get the DAG from the hydrolysis of phosphatidylcholine and the phospholipid from the membrane, and also hydrolysis of phosphatidylcholine will persist for hours. Okay, instead compared to the phosphatidylinositol biphosphate PIP2. The response of PIP2 is very transient, while the response of another type of phospholipid like a phosphatidylcholine is longer, persists longer. So this one is very important for the long-term response. Okay, this is the end of my afternoon lecture today.